we present Vimo, a visualization tool to search and analyze connectivity motifs in large brain networks. Neuroscientists can reconstruct three-dimensional models of neurons using high-resolution electron microscopy to better understand how the brain processes information. For example, here we show a set of neurons in the central brain of the fruit fly. Those neurons have complex three-dimensional shapes and form large connectivity networks. But what do we actually mean by neuronal connectivity motifs? Brain networks can be interpreted as directed graphs, where nodes are neurons and edges are connections between neurons, so-called synapses. Motifs are small repeating subgraphs that can be seen as building blocks of computation in the brain. For example, new research shows that the specific motifs in the fly brain mediate responses to temperature. But finding and analyzing these motifs is inherently complex. The most recent connectome datasets contain more than 50,000 neurons and over 100 million synapses. For such large graphs, motif discovery is computationally expensive and typically requires writing code in complex query languages. Also, scientists want to explore not only the high-level connectivity between neurons, but dive deeper into how neurons actually form a motif in 3D space. However, the three-dimensional structure of individual neurons is complicated, and neurons are often very entangled. That makes it difficult to mentally map the graph structure of a motif to the actual 3D morphology of neurons. Vimo addresses these challenges by allowing users to interactively search and visualize arbitrary connectivity motifs in large brain networks. Users can sketch a motif and define biological constraints like neuron types and connection strengths. Motif constraints make queries more biologically meaningful and also reduce the search space of the motif discovery algorithm. Here we show the motif drawn in task 1 of the case study in the paper. For each sketch motif, we also display how often it occurs and if it's over or underrepresented in the data. As a next step, users search for instances of the sketch motif. The list view shows found instances, and users can select and inspect the results in more detail. To better understand how neurons form a motif, we've designed a novel focus and context approach that gradually highlights the connectivity in the three-dimensional dataset. First, users can slowly prune all neuron branches that are unrelated to the motif, highlighting the core motif connectivity. Second, by dragging the slider further, Vimo untangles these neurons by transitioning to an exploded view. This allows users to inspect complicated neuron morphology with less occlusion. Third, we hierarchically cluster and bundle connections between neurons to highlight synapse clusters. Additionally, Vimo allows graying out unrelated neuron branches and highlighting the brain regions to provide spatial context for neuroscientists. Neuroscientists also use Vimo to study the spatial relationships between different motifs instances to get a more holistic view of information processing in the brain. On the one hand, Vimo supports a neuron-centric analysis by showing if a neuron forms the same motif with different synaptic partners. On the other hand, users can also explore synaptic pathways starting from a seed motif instance. They can trace pathways built by repeating occurrences of the sketch motif by setting specific neuron IDs as node constraints before querying the brain network again. In our user study, we have shown how our tool lowers the threshold for neuronal motif analysis, and we hope the neuroscience community will adopt Vimo into their workflow.